Hoy día les vamos a presentar, le quiero presentar al doctor Kartik Chandran. Él es un investigador, pertenece al Albert Einstein College of Medicine. Hemos estado trabajando con él, me avisé que iba a hablar en español. Hemos estado trabajando con él en un proyecto que se llama Prometheus, hace 5 años, que reúne un consorcio de investigadores de Finlandia, Suecia, Estados Unidos y Chile, y que tiene el propósito de identificar las áreas de virus emergentes que son susceptibles de ser atacadas con anticuerpos monoclonales en forma específica. Y afortunadamente, gracias al esfuerzo de Kartik, este año se presentó un nuevo proyecto al NIH. Son proyectos muy grandes en que participan el mismo consorcio de investigadores y fue financiado un proyecto que se llama Providence, así que tenemos trabajo para los próximos 5 años de altísimo nivel, como ustedes van a ver con ellos. Algo nos llega y algo crecemos con esta relación. Kartik es un investigador que se ha dedicado toda su trayectoria científica a estudiar la interacción inicial que tiene un virus con las células y al desarrollo entonces de tratamientos que bloqueen esa interacción. Los virus específicamente que ha atacado son el virus ébola, exitosamente con desarrollo de anticuerpos monoclonales, el SARS-CoV-2 cuando surgió la pandemia, que surgió en el medio de este proyecto que tenía con el NIH y en la cual participamos, y también que era la parte que hace con nosotros, hantavirus. Y finalmente explicarles el título, porque la verdad es que no lo quise traducir al español porque Kartik hizo un juego de palabras. I'm explaining the title, spiking. Como ustedes oyeron innumerables veces en coronavirus, hay una proteína spike que sobresale, presenta al virus ante el sistema inmunológico y la interactúa con los receptores celulares, y al mismo tiempo spiking significa rematar. Entonces aquí hay un juego de palabras entre rematar y usar la proteína spike de la superficie del virus para en esta infección a través del uso de anticuerpos monoclonales.
Voleibol, ese es un spike. Yo soy muy buen voleibolista por mi altura.
Ok, bueno.
That pun really doesn't work in Spanish, I guess.
Okay, so Kartik, it's an honor to have you here. He came for the Congreso Futuro. He gave a very nice talk, very motivating talk. The Ministry of Health was in the audience, and he got to hear a very nice message on what we have to do on research and clinical work for the hantavirus infection here in Chile. So thank you very much for being here, for taking time to come to the university. We're very honored with your presence here, and now the floor is yours.
I'm going to trip over this chair. It's a real pleasure to be here. Thank you, Pablo, so much for inviting me to speak. I've been in Chile since Saturday and just been exploring the city and having a really good time. A really beautiful country and a really beautiful campus as well. So as Pablo said, today I'm going to be telling you about this work that we've been doing together as part of this bigger consortium called Prometheus, and I'll introduce all of that to you. to develop antibodies against emerging infectious diseases. And one of our special focuses of this grant is to do this, to develop human monoclonal antibodies that we could use as broad therapeutics to treat hantavirus infections, not just here in Chile and in South America, but also in the US and in Europe, where there are different kinds of hantaviruses that jump into people and cause human disease. So for me personally, a lot of this work really comes from the work that we started when I started my lab about 15 years ago now, working on Ebola. And when I had initially, done my work on Ebola, it was this virus that really nobody cared about. It was the people that cared about Ebola, had read The Hot Zone, which is this very popular book about Ebola outbreaks, but it wasn't really considered a significant public health threat at that time. And I started working on it really because I was just curious about the interesting biology of this virus. I wasn't thinking about doing anything translational to, beyond just basic research. In fact, a lot of public health sort of nonprofits like Gates Foundation didn't really want to fund any work on Ebola because they're like, this is not that important. We're solving much bigger problems like HIV and, maternal health and all of these things. And of course, at that time they were right. But what happened that nobody expected, that nobody anticipated, and that's how emerging infections are. Nobody sees it coming till it happens, like COVID-19. There was this enormous outbreak of Ebola in West Africa between 2014 to 2016 in a part of the continent that had never seen these viruses before. For the first time ever that we know about, Ebola spread to densely populated urban areas and essentially infected thousands of people, killed more than 25,000 people. So, and just as important, it absolutely destroyed all of the work that Gates Foundation and other health agencies and nonprofits had done to improve human health, to create infrastructure, to train people, because so many doctors and nurses died. And the distrust between the population and the government, the population and the medical establishment, just became very, very significant during that outbreak. And of course, there were huge effects on these economies as well. So I think for us, the lesson that we took away from it is that just because a virus is not considered an important public health concern today, it may not stay that way. That virus or a virus related to it may cause an outbreak. And as the world is changing and as the climate is changing, as humans become even more interconnected, the potential for a virus to spread locally, regionally, and globally keeps increasing. And so I think the WHO took as well that outbreak to heart and came out with this blueprint for preparedness and response for emerging infections. And this is kind of where our work started, to really go more from just the research bench to really think about a more translational effort to develop therapeutics. And so in that outbreak, there were many, many people that became infected and survived. And so we were fortunate to be able to be part of a collaboration to isolate monoclonal antibodies from somebody that had recovered from Ebola virus infection, an American doctor. And from this person, we isolated 400 monoclonal antibodies. And just through a process of the study and mining, really, in a way, we were able to identify 2 monoclonal antibodies. Don't worry about the names of the antibodies, at least these ones. So what we came up with in the end was a 2-antibody cocktail of these 2 monoclonal antibodies. And so these are shown in pink here, and they're bound to the spike glycoprotein of Ebola virus, which is sort of coming out of the screen in this view. So they kind of bind on the side at the same time. And so what we were able to do is start with these 2 fully human monoclonal antibodies, and then engineer them to make them bind even better and more broadly to different kinds of Ebola viruses. Because everybody thinks Ebola virus is just one virus, but just like as hantaviruses are, there are multiple sort of Ebola viruses out there in nature that can cause human outbreaks. And all of the antibodies that had really been characterized previously were specific for one kind of Ebola, but we wanted something broad. And so we were able to engineer the antibody to broaden its affinity and its breadth. And I'm sort of giving all this as prologue because a lot of the work that Pablo and I and our big consortium have done on hantavirus is really building on what we learned from these studies on Ebola. We also engineered the FC domain of the antibodies, which interfaces the antibody to the immune cells to affect killing of infected cells, to make that process of cell killing even more efficient. And this resulted in a clinical product that is incredibly effective in treating Ebola virus infection in non-human primates. And so what I'm showing you here is 3 different models of Ebola virus, lethal Ebola virus infection in non-human primates. 3 different kinds of Ebola virus, Ebola, what's called just Ebola virus, but then also used to be called Ebola Zaire, for those of you who are interested in these viruses, Bundibugyo virus, which is another Ebola virus, and then Sudan virus, which also causes human. All 3 of these viruses cause human outbreaks. That's the big one, but these guys also are quite important. And so these are all lethal models. You can see from the black lines that if you don't give any treatment to the non-human primates, they all die. But we can give, and this is an older experiment, so 2 doses of antibody, or in these 2 models, a single dose of antibody, and get almost complete or complete protection. And an important point here to mention is that the antibodies are not given until the animals are already viremic. So they already have a fever, they're already making virus, and they already have significant tissue injury in the liver and in other organs. And we can actually completely reverse all of these things. We can reduce the viremia down to 0. We can reverse liver injury. We can reverse inflammation. And so this is really the first time that anybody was able to show that a single dose of a monoclonal antibody could be given at a late time point in infection in this incredibly lethal sort of disease models and get protection. And so we, based on all of this work, we were able to convince the US government to fund the preclinical development of this antibody cocktail and actually take it all the way through to clinical grade manufacturing and fund a phase 1 clinical trial, which just finished. And so now we have a phase 1 clinically approved, a safety approved product. And now, of course, the problem is, who's going to pay to make a bunch of it and keep it in the freezer in case there's another outbreak? So that's what we're working on. And you'll see that this is sort of a repeated theme with these emerging viruses. It's very difficult to get money to carry it through all of the processes that are needed to actually get a product that can be used in people. And then what do you do with that product? Can you go and do a clinical trial for efficacy and get approval, or do you just keep it in the freezer and wait? So this is sort of like a discussion maybe we can have at some point. So based on all of this work on Ebola, that really drove us to create a consortium that we call Prometheus. And for those of you who are not familiar with Prometheus, he's basically a character in Greek mythology who's a human that stole fire from the gods and brought it to the people. And of course he was severely punished for it, but that's not the point of the way we're using that term. Prometheus literally means looking ahead, and so that's sort of how we think about our consortium. We're trying to anticipate a future need rather than something that maybe the consensus is, hey, this is not a problem right now, but we think it could become a problem in the future, and so we're trying to develop and pre-position these antibody therapeutics for future use. So this is a large consortium with lots and lots of different investigators all over the world, and of course, Pablo and Cecilia Vial are a really crucial part of our efforts, but also investigators in Sweden, and in Finland, and in France. So it's like a really international collaboration. Also has academic institutions like ours and UDD, but also industry, so companies that are like helping to do the discovery, but also translate the findings, and then government institutions that are playing a key role in doing a lot of the animal studies that I'm going to show you today. Okay, so that was just a prologue about Ebola, which really set the stage for our work on hantaviruses, and really the rest of the talk I'm going to tell you about hantaviruses. This audience, I don't need to belabor a lot of this, so you know that hantaviruses are this incredibly diverse group of agents, and the mammal infecting members of this family can infect lots of different kinds of hosts, even these really weird fish-looking things. This is called a beanie pizza batfish that swims in the South China Sea and has a very divergent virus that can be considered a hantavirus. As you know, of course, there are these 2 major disease syndromes that hantaviruses cause: HFRS, hemorrhagic fever with renal syndrome in Europe and Asia, and in North and South America we have HCPS and especially Sinombre in the US and Canada, and Andes virus in Argentina and Chile. And of course, this is all review. I mean, I've given this talk at places where people don't know what hantaviruses are, so this is clearly not this audience. But and then of course, as you are all quite familiar, there are no known intermediate hosts for hantaviruses that cause disease in people. They seem to jump directly from their rodent or mammalian hosts to humans just through fomites and sort of direct exposure. For the purpose of this talk, maybe the more important point is that there are no approved vaccines or therapeutics to treat any of the hantavirus diseases. And we know from studies with old world hantaviruses like Pumala that low antiviral antibody titers correlate with poor disease outcomes. So with Pumala virus, our collaborators in Sweden have shown that individuals that present at hospital with more severe disease are more likely to have lower antibody titers. And then of course there's studies from Pablo and his collaborators showing that convalescent plasma has therapeutic benefit against Andes virus, implying that antibodies that are in these convalescent plasma can have an antiviral effect and provide therapeutic benefit. So the question that Prometheus asked was, could human monoclonal antibodies that we isolate from survivors have therapeutic utility, not just against one group of hantaviruses, but against all of them? At least the ones that we know about that cause disease. So a little bit of virology. This is just a really nice image, reconstruction of what a hantavirus particle looks like from Pablo Guardado in Felix Rey's lab at Institut Pasteur. And so what's really interesting, a couple of interesting things about these glycoprotein spikes is that they're tetramers, unlike a lot of other spikes for lots of other viruses like COVID or Ebola or HIV, which are trimers. And then they also make these, they interact laterally to make these rafts, but the rafts are not these completely closed lattices like you see with dengue virus or something. They sort of have like these bald patches, like clearings in the forest where there don't seem to be a lot of glycoproteins, which is another really interesting feature. And I'm bringing this up now because we think that the organization of these particles has implications for the ways in which our monoclonal antibodies work. So then, of course, there's a viral membrane, the envelope in which these glycoproteins are present. There's a trisegmented negative strand RNA genome that encodes the viral proteins. And then the key protein for this talk is the glycoprotein complex GNGC, which has this unusual tetrameric architecture. And so we now know a lot more about the organization and this defined structure of this glycoprotein complex, especially thanks to work from Felix Rey's group at the Pasteur, who was part of Prometheus. And so basically what they showed is that, well, first of all, we know that the glycoprotein is made as a single polyprotein that's proteolytically cleaved into GN and GC, and there's this intermediate NSM protein that's not in the virus particle that's sort of cleaved out. If you look at how this is organized in this kind of two-dimensional diagram, what we now know is that the N-terminal subunit of the glycoprotein complex, which is called GN, forms this head domain that interacts closely with the ectodomain of the GC subunit shown in yellow in a one-to-one interaction. And then there's a base domain which then leads into a series of transmembrane domains to make the folded structure. And the tetramerization domain is down here. So what Pablo was able to do was solve the crystal structure of just this part of the glycoprotein, which is the ectodomain that has the GN head, which is shown here as GNH and in red, and the GC, the entire GC ectodomain going into the transmembrane domain, which is shown here. And so for those of you who are really interested in viral membrane fusion, you would see that the GC subunit is really what you would call a class 2 fusion protein. In other words, it looks like the fusion glycoproteins of dengue, like flaviviruses like dengue and alpha viruses like chikungunya virus. So it has the same kind of structure and architecture and presumably works very much the same way. Importantly, for this particular virus, the GN head domain interacts really closely with the GC subunit. And at the top of the GC subunit, you have these hydrophobic loops that are called fusion loops, and I'll show you on the next slide. These are the loops that insert into the target membrane during membrane fusion. So the GN plays a really important role here in kind of holding and covering these GC subunits and holding the GC fusion loops and holding the GC subunit in a pre-fusion conformation. And then when the pH drops in the endosome, GN lets go and GC rearranges to cause membrane fusion. And so this is just, I'll show you a couple more slides of that. This is just fitting this monomer of the GN and GC ectodomain into this tetrameric lattice, which works really beautifully. And so this is the top view and this is the side view. The side view is really fascinating because it looks like a carousel. I don't know what the Spanish word for is that. Okay, perfect. So this is sort of like a carousel, and the GN part makes the sort of central canopy that spins around, and then the GCs are like these horses, that are like hanging off the sides of the carousel. And so this is what the structure actually looks like in the intact virus particle. So one kind of complication of this beautiful but very kind of, complex structure is that nobody has yet figured out how to make a soluble tetramer that we can just produce in the lab. And we can make, the proteins I'm showing you are just monomers. This is not a real structure. This is just a fit of the monomeric GNGC complex into the tetramer. So nobody's actually made this structure as a soluble protein, which is a problem for us to isolate monoclonal antibodies because we need a soluble protein to really do this well. But I'll show you that we've come up with a hack to solve this problem in our antibody discovery efforts. So just going back to the mechanism of infection, essentially this is sort of a cartoon and a side view of the glycoprotein complex. So what we believe happens is that the virus binds to a receptor. We've identified a receptor for this virus in the last few years called protocoderin-1 for Andes and Sinombre virus. And that we believe binds to the GN subunit, although we haven't completely mapped that. And then the virus is taken up into endosomes where the pH gets more and more acidic. And at acid pH, this whole complex dissociates. And the GC subunit refolds to make these trimers that insert into the target membrane and then fold back on themselves to cause a fusion of the viral and cellular membrane. And so what you end up with in the end is a trimeric structure which looks exactly like the post-fusion trimers of alphavirus and flaviviruses and even rubella virus. And there are even some cellular proteins, so this is from a worm, that is a cellular fusogen that makes the same kind of structure. So basically, this is a very ancient way to fuse two membranes together that's mediated by the GC subunit. But hantaviruses evolved to take this GC subunit that has a very sort of common mechanism and adapt it to a completely different purpose, which is to get the virus to fuse with the membrane of the cell. And the way that it's organized is totally different because it's a tetramer and it has this other piece called GN, which all these other proteins don't really have. But nevertheless, the actual fusion mechanism, once it starts, is probably quite similar to all these other viruses. Okay, so with that as background, let me tell you a little bit about how we go about isolating these monoclonal antibodies. Our platform really relies on human convalescent donors, and so the really key roles that some of our collaborators play in this is providing these validated peripheral blood mononuclear cell fractions, or PBMCs, from individuals that have been exposed to and recovered from infection. And so we have samples from Pablo for Andes virus. We have samples from Stephen Bradfield at University of New Mexico for Sinombre. And then we have samples from Umeå in Sweden for people that have been exposed to Pumala virus. And so these are the three sort of human sets of samples that we're using to isolate our monoclonal antibodies. Once we've got these PBMCs, we need a way to go in and fish out only the B cells that are displaying the right B cell receptors for the antibodies that we want, right? And so we need, if you want to go fishing for a certain kind of fish, you need a very specific bait just for that fish. And so we use fluorescently labeled proteins as bait. And actually, in the case of hantaviruses, we're using a virus particle, and I'll show you how we do that on the next few slides. And so by using a specific bait and this PBMC fraction, and we can FACS sort for viral antigen-specific memory B cells. And these B cells are then single cell sorted into 96 well plates. And then this is all pretty standard methodology that lots of people do. We do use nested PCR to amplify the variable heavy and light chain sequences as PCR products from these single B cells. And this is where things get a little bit more sort of interesting and efficient. Instead of taking these PCR products and cloning them into mammalian expression vectors and producing antibodies in mammalian cells, which of course you can do and lots of people do, but it's kind of slow when you've got hundreds of antibodies. Instead of that, what we do is we use recombinational cloning to display these antibodies on the surface of yeast cells by yeast display. So I don't know if people are familiar with yeast display, but you can actually display proteins on the cell wall of yeast in a very specific way. And so you just engineer them ahead of time to display an antibody. And so then all you have to do is transform this yeast strain with your variable heavy and light chain antibody products, and it all just goes right in like magic, and you get an antibody on the surface. And we can then use these displayed yeast to find only the antibodies that are binding to our hantavirus glycoprotein. And then we can sequence them and sort of like kind of analyze all of the different antibodies that we find. We can group them by germline and do all these sorts of things. We can also sort of do some computational analysis for what the industry people like to call manufacturability or developability, because all these antibodies that you pull out of natural repertoires aren't always suitable for large-scale production. They may have some sequence liabilities. These can be fixed at this stage. And then one of the really cool aspects of the system is that just by adding a small molecule to the yeast culture, we can get the yeast to secrete the antibody instead of displaying it on the surface. And so this is good enough that we can actually get enough antibody to do binding studies by biolayer interferometry, and to do viral binding and neutralization screens to find antibodies that neutralize or block infection. One of the enabling sort of technologies for all of this work is a viral entry platform that can be used in BSL-2, so that we don't have to do work with authentic viruses, authentic hantaviruses at BSL-3. There's no reverse genetics for hantavirus, as I'm sure you all know. So it's not possible right now to make a hantavirus with a reporter gene like GFP. So then it's just a pain to do anything, because you have to do immunostaining for everything. And it's a lot faster to screen things in BSL-2. So this is kind of where one of the strengths of my lab comes in, which is our ability to build recombinant VSVs or vesicular stomatitis viruses, displaying different spike proteins as a BSL-2 model for lots and lots of different viral glycoproteins. Again, I'm sure you're all familiar with these VSV pseudotypes, where you basically you kind of introduce a foreign spike protein into the VSV particle, and you use that to do neutralization assays. I think what we do is we take that one step further. We actually genetically engineer the genome of the virus to replace its own glycoprotein, which is called G, with that of any virus of interest, in this case, hantavirus. And we can also put any reporters we want, like GFP, which makes it a lot easier to measure infection. But one really powerful aspect of this, putting the glycoprotein into the genome, is that this is actually a replicating virus. And since this is an RNA virus, it undergoes mutation at a high rate, which means that this virus will exist as a quasi-species that includes sequence variation in the GP gene, which means we can do forward genetics. And I'll show you that we can use it to select escape mutants to antibodies, which is a very useful way to map where the antibodies bind and do other types of functional studies. And if you feel like traveling down to University Católica on Friday, I'm giving a talk there about our work on coronaviruses, which really leverages quite extensively the ability to do forward genetics with the VSV system. So one question we have when we're starting to work on these viruses, because these glycoproteins have these unusual tetramers, we wondered how they would be distributed on the VSV particles. And so we actually collaborated with Jason McClellan's group at UT Austin in Texas. And what we found is something really, really unusual that hasn't really been described previously. So I don't know how well you can see this, but I actually marked this. If you look at where the hantavirus glycoproteins are on the particles, so just to back up a second, VSV makes these bullet-shaped particles. So there's a curve, just like a bullet. There's a curved front end. There's a more flat back end, and then the sides are just a cylinder. And what we find is that the hantavirus glycoproteins are all clustered at the tips. They're not everywhere. In general, the sides are relatively deficient or denuded of glycoprotein complexes. This is in contrast to a VSV that's expressed in the SARS-2 spike, where it's a bit harder to see that, but because the glycoprotein isn't as big, but it's all over the particles. And that makes sense intuitively because these are trimers that don't really interact laterally. They just sort of like are independent. And so it makes sense that they can go everywhere. But we think that with hantavirus, because the glycoproteins make these tetramers that actually make lattices, that probably influences budding. And so these glycoprotein lattices like to exist in areas of membrane curvature. That's our theory. And therefore, they're sort of concentrated at the tips. And this sort of creates some interesting, we believe that this has consequences for the behavior of some of our monoclonal antibodies, because even though and then I should point out that Jason's group, postdoc Luqiang Guo in his lab, has actually generated a relatively high resolution reconstruction of the glycoprotein lattice from VSV particles. So what you're looking at is the EM density from the VSV particles, and into that has been fitted a tetrameric model from real hantavirus. So the VSV actually displays authentic looking spikes and authentic looking lattices, but the way the lattices are put together at a higher scale of organization is not the same. It's on the tips, it's not all over the particle like you see with hantavirus. So that's sort of interesting from if you're a virus nerd, but also something to keep in mind when you're thinking about developing therapeutics and the consequences this sort of organizational differences might have. People always ask me this question of what does a glycoprotein look like in VSV? So I'm finally happy to have some data on that. Okay, so just on to the antibody discovery efforts. We initially actually started out with pumila donors from Sweden, and it was just because that's just what we had initially, and they had a lot of samples. And we're also excited because pumila sort of sits in an intermediate position between the old world and the new world clades, so that we were hoping that maybe this would mean we would get antibodies that could recognize both new world and old world hantaviruses. That turns out to be the case, but probably not for the reason that we thought anyway. And in fact, we could get antibodies out of Andes virus convalescents that can also bind pumilavirus glycoprotein. So anyway, nevertheless, it was an initial thought that pumila might be a good place to start. So then we had to solve this other problem of how to actually make a fluorescent bait to go fishing for B cells. And so what we ended up doing was to actually engineer a new VSV that displays the pumilavirus, expresses the pumilavirus glycoprotein, but has a fluorescent protein, amnion green, fused to the viral phosphoprotein. So in the previous virus that I showed you, we just have GFP as a free-floating protein, so it doesn't really efficiently incorporate into virus particles, which is what we needed here. So by making this P fusion, which is actually packaged into the virion, we can actually get essentially fluorescent viral beads that we can use. And this actually works quite well. It's not as good as a recombinant protein, but it does work pretty well. So what you're looking at here is a flow plot showing the amnion green fluorescence of gating on CD19-20 positive B cells. And these are actually kind of double labeled. And so what you're looking at here is in this sort of boxed quadrant, these are the B cells that are positive for binding to this recombinant VSV. And so this is a control donor. There are some events. It's a virus, it's a little sticky. So we're getting some background. But you can see a lot more positive cells from the convalescent donor. So that was really encouraging because it really suggested that we are finding authentic B cells that are authentically expressing a pumilavirus glycoprotein specific B cell receptor. And that turned out to be the case. And after some work, we were able to identify about 180 monoclonal antibodies from 3 different donors from Sweden. And then we did a whole bunch of screening, which I'm going to summarize 6 months of work on like 2 slides. So what we're really looking at here, and so the other thing that we can do really nicely with the VSV system is build VSVs with different glycoproteins in them from different hantaviruses. And so what you're looking at here are viruses that are both old and new world, and we can use exactly the same assay to screen everything because all of these have the same reporter. This is a heat map looking at neutralization potency for these 5 different viruses, going from non-neutralizing to highly neutralizing, like less than, this is actually in the picomolar range for neutralization IC50 in tissue culture. And then these are all kind of like ranked according to breadth. So again, from the very beginning, we just didn't want a pumalovirus antibody because, you know, I don't know, I mean, we can't make a different antibody for each of these viruses. There's not enough money in the world to do that. So our only shot of getting a therapeutic would be to get something broad. So that was sort of a thought from the get-go. And that's why we initially just triage based not just on potency of neutralization, but also breadth of neutralization. And that sort of gave us this set of antibodies here on the left. Then we took you know, these antibodies and some other ones as well, and then did some epitope mapping. And so we did this different ways. I'm just showing you one way that we did it, which was to actually take some of these antibodies and select escape mutants with the VSV system. And what we get are, and I'll tell you a little bit more about some of these, we get escape mutations in the GN subunit as well as the GC subunit with different antibodies. And then what was really nice was that we could take that virus that had the escape mutation to one antibody and then test it against all the other antibodies. And some of the other antibodies would also, you know, not be able to neutralize that escape mutant virus, which told us that they share the same epitope or very similar overlapping epitopes. And so that was one, allowed us to sort of cluster antibodies in terms of GN binding based on escape in GN, and then and GC binding based on escape in GC. And then we had these other antibodies that seem to have more complex epitopes for the most part, but really none of these were broadly neutralizing. They were all Pumala specific, so we just sort of, we put them aside for now, and when we have bandwidth, we're going to go back and look at them a little bit more. So I'm really going to focus on these two groups of antibodies. And so then, of course, our next question was, we have a rough idea from epitope mapping where they bind in GN or GC, but can we show structurally where do they bind into this lattice of the hantavirus tetramers on the particle? And so the first way we started out by doing this, and this is all work done by Pablo Bordado in Felix Rey's lab, who now has his own group at the Pasteur working on pox viruses. We're also working on pox viruses, so yeah, because big DNA viruses are really interesting. Lots of genes to mess with. But anyway, this was when he was still in Felix's lab and working on hantavirus structures. So what Pablo did was he basically did a negative SANE EM on this GN-GC monomer that I showed you, the heterodimer, which is just the head part of GN and the whole of GC ectodomain, bound to Fabs of the antibody. And so this is the GN-GC and this is the antibody. And then he was able to actually make some class averages of these EM structures and then very nicely fit in the crystal structures of the GN-GC and a generic antibody. And what this showed us was really sort of unexpected was for some of the group 1 antibodies, so remember that these are antibodies that escape by mutations in GN, but the way they bound or seem to bind to the glycoprotein is really to an epitope that seems to be very close to an intersubunit region. So not just a purely GN contact like on the top of GN, but really something that's more on the side or kind of at an oblique angle here, as you can see, that seems to be close to an epitope, close to sort of where GN contacts GC in the structure. In contrast, the group 2 antibodies are definitely GC binders. They bind more to the side, as you can see from the side view, and they're definitely just contacting GC and not contacting GN, which makes total sense because that's where the escape mutations are as well. And I'm not going to talk more about this, but one of the interesting little side note here, which goes back to those VSV structures that I showed you of how the glycoproteins are distributed. These antibodies are really good at neutralizing VSV, but they're really terrible at neutralizing authentic hantavirus. And we think the reason for that is because the VSVs have these more incomplete lattices with more spaces where the antibodies can kind of get in on the side. But in authentic hantaviruses, you have much more fully formed these tetrameric lattices, and the antibody may not be sterically inhibited from getting in and binding on the side. Anyway, that's just a working hypothesis. We don't actually have direct evidence for that yet. But the group 1 antibodies seem to work equally well with VSV as well as authentic hantaviruses. Once we had sort of some data on these antibodies, we wanted to very quickly triage them for protective efficacy in some kind of animal model. And the most conservative thing to do was to use a pulmonovirus model, since that is the virus against which these antibodies were elicited. And so we collaborated with Matthias Forschell's group, and with, who's at University of Umeå in Sweden, and his student Jana Tinnell, who's actually working in Thomas Stranden's lab at University of Helsinki in Finland. And they have this really novel and interesting model where they can infect the bank vole, which is actually the host for pulmonovirus. So they can find wild-caught bank voles that are actually not infected with hantavirus. Many of them are, but some of them aren't. And then they can essentially give them this strain of pulmonovirus that they found that grows pretty well in the bank vole. And then they can measure viral load by doing qPCR for different genome segments. And so this is just an example of what happens in different tissue with the pink circles. If you don't do any antibody treatment, you get nice load in different organs, especially in the lung. And if you pre-treat these bank voles with different monoclonal antibodies that we isolated, you get really nice protection. So the antibody essentially prevents acquisition of pulmonovirus infection in these animals. We can also do a more stringent experiment where we infect first and then give the antibody. Still not super stringent because we're giving it like the next day. But still, it's a more difficult scenario for the antibody. And sure enough, we don't see as good protection. So we do see in the lung especially significant viral replication in the presence of antibody. However, we do see a very nice reduction of anywhere from 100 to 1,000-fold in load. And then in some of the other tissues where there isn't as much replication, we see essentially like no evidence for viral replication or below the limit of detection. And that's quite interesting that in serum as well, we see this, which suggests that the antibody might be preventing dissemination of the virus in the bank voles through the blood. By doing some more experiments that I'm not really going to show you, we kind of narrowed everything down to a single antibody, which is a group 1 antibody that kind of we thought bound to this intersubunit epitope between GN and GC called 42898. So if there's one 5-digit number to remember, I'm going to ask you to remember this one. But it's the only one because I'm going to be talking about it for the rest of the time. And so this antibody really stood out because it seemed to work the best against both old and new world hantaviruses. And so that's the one we proceeded with. And then, of course, the real acid test here is, does it work against Andes virus in the Syrian hamster model? And the answer is it does. So, so these are experiments done by Andrew Herbert's group at USAMRIID, which is a part of the US Army outside of Washington, D.C., in high containment. And so what they did was expose Syrian hamsters to Andes virus and then come in 3 days later with with 2 different doses. So a single dose, but a single sort of administration of antibody, but a high dose and a low dose. And then this is a pretty lethal model, sort of uniformly lethal, especially intramuscular with intramuscular infection. And so you can see that all the control animals die, but all of the antibody-treated animals survive. So this was really exciting for us. And this correlated with a really nice reduction in viral load in the lung to below the limit of detection. So the antibody works. I mean, obviously, giving the antibody 3 days post-infection is not very realistic in any kind of real clinical setting. But this is the first test that the antibody has to pass. And if you look at the published literature for antibody therapeutics against hantaviruses, this is the kind of experiment that's typically done. It's a pretty low bar for protection, but that's where we started. Towards the end of the talk, I'll show you some more stringent tests for protection in the Syrian hamster model. Okay, so just going to spend a little bit of time talking about mechanism. Probably you'll let me know when I'm running out of time, because I'm not quite sure when I started. So our question was, how does this antibody 42898 recognize the glycoprotein complex, and why is it broadly neutralizing and broadly protective? We really want to get at the mechanism of this. And so through a collaboration with Pablo and Felix and a postdoc in my lab, Eva Mittler, we were able to solve the crystal structure of this heterodimeric GN-GC complex in complex with a single chain FV of our antibody of interest. And just to orient you, here's a GC subunit shown here vertically, and the fusion loops that are kind of going to contact the membrane during the fusion reaction are up here, BC and CD in this rectangle. And you can see this pink loop that's sort of inserting itself here. That's called the capping loop, and it's coming from GN. The rest of GN is sort of in the back, is behind the screen here. And then you can see the antibody here in green. And so you can see that, as the EM suggested, the antibody does contact both GC subunits, the fusion loop specifically, and the GN subunit, and this one particular pink capping loop. And so what you're seeing here is really just a blown up version of this interaction. I'm not going to go into all the structural details, but the main point to make here is that here's the viral GC fusion loops, and so the one of the antibody chains really contacts these quite extensively, and then here's the GN capping loop, and there are also contacts with the light chain of the antibody with the capping loop. So this truly is an intersubunit epitope, with the light chain making most of the GN contacts and the heavy chain making most of the GC contacts. And this is the first time that this type of antigen-antibody interface has been described for a hantavirus, or really any kind of bunnivirus. This is just a different view of this, which kind of makes two different points. So what you're looking at in white and purple is the surface of the GN-GC complex, and the green is the outline of the antibody bound to this complex. This is like, when there's somebody gets killed on the street in their bodies and they do a chalk outline, it's a little bit like that. And so the colors have to do with sequence conservation. So purple means highly conserved, and white means not conserved. And so you can see that the epitope is quite interesting because it actually straddles a region of high conservation, which is the GC fusion loops, these are highly conserved, and a region of high variability, which is this capping loop. And that's really kind of weird and unexpected, or it was to us, because if this is a broadly binding and broadly neutralizing antibody, you would think that the entire epitope would be highly conserved, but that's not what it looks like. But we think we understand how this works, and I'll show you that in the next few slides. This is just showing you again a little bit that, again, here's GN and here's GC, so you can see how the antibody is really just, truly kind of stapling together the two subunits. So if you look at the sequence conservation of the two major sets of contacts in the GN subunit, which is this capping loop, and the black ovals here are just showing you the contact residues in the virus to the antibody, and here is the fusion loops in the GC. You can just see, just by looking at it, that GN is way less conserved than GC, and in particular, the critical contact, or the main antibody contacts in the structure are all really quite variable in the GN. I'm just showing you the sequence of what I showed you on the previous slide. In contrast, a lot of the critical contacts in GC are quite conserved. So how does the antibody able to broadly recognize and neutralize hantaviruses if in fact it's making contacts with this highly variable capping loop in GN? And the answer seems to be that when you look at the structure, most of the contacts of the antibody side chains are not with the side chains capping loop, they're with the backbone of the capping loop. So there's only one residue that really seems to be quite important, which is this glutamine in making a side chain contact. And sure enough, when we select VSV, Pumila VSV that can escape the antibody, this is a residue that mutates. So that residue seems to be quite important. But a lot of the other hantaviruses don't actually have a glutamine at that position. They have an asparagine or a leucine at that position. So even though that contact seems to be important, it's not indispensable because the viruses can still bind to the antibody. The antibody can still bind to these divergent viruses that don't have a glutamine at that position. So then the question arises, what happens when you select escapes to, say, Andes virus, which has an asparagine there instead of a glutamine? Can the virus escape, this antibody at all? Because if you're contacting only the backbone, how do you escape it? So we thought initially we would get GC escape mutations, but that's not what we got. And we think we don't really get GC escape mutations very easily because GC is functionally very conserved, and so there's probably a higher fitness cost to mutate GC. What we saw was something we totally did not expect, which was really kind of fun, which is that all of the mutations that we saw essentially create glycosylation sites in the capping loop. So they essentially create NXS or NXT sites that are where the asparagine is subject to N-link glycosylation. And so for all of the viruses that don't have a glutamine at that position, and so essentially what we think the virus is doing what antigen and protein engineers do, which is to resurface parts of the protein you don't want antibodies against by putting sugars on it. But the viruses just figure out how to do that on the capping loop just by adding sugars. So I think that's kind of neat. We would be really interested in looking, and I think this is something that Pablo is working on to to look at actually the sequences of these viruses in people to see if these kinds of mutations arise. And so far, the answer seems to be no, right? Or not? It's a work in progress. Yeah. Okay. So that's sort of the structure of how the antigen contacts the antibody contacts the antigen. So something that we care about a lot in the lab is mechanisms of viral entry, and so how antibodies actually block viral entry. And we know it's entry because it works in the VSV context, which is really only just measuring viral entry mediated by the glycoprotein. So just to set up the slide, our working hypothesis was the way the antibody binds by stapling GN and GC together, and the fact that we know that the complex has to dissociate to allow the GC fusion loops to insert in the target membrane meant that a really plausible mechanism for the antibody is that it just holds the subunits together and keeps them from coming apart. And so it's really just acting like a staple. So to test that directly, we wanted to measure the effect of the antibody on viral membrane fusion. And there are different ways you could do this, but the way we decided to do this was using what virologists like to call a fusion infection assay. And so what you do here is you essentially force the virus to fuse at the plasma membrane instead of fusing out of an acidic endosome. And you do that by treating cells with ammonium chloride, which neutralizes the pH in endosomes. So the virus is basically dead, it can't enter through the normal endocytic pathway. And then what you do is you bind the virus at 4 degrees. So it's just stuck to the plasma membrane. And then you just do a quick pH temperature shift. So you just add media that's been titrated to whatever pH you want at 37 degrees for like 2 minutes. And then you replace the media again with media that have ammonium chloride in it. And then you let the infection go, and then you score for infection the next day. And so what you're looking at here is really just a pH curve of infection, where we clamp the pH of the extracellular medium to different values. So of course, with no virus, you get no infection because there's no virus. And so you're really measuring GFP here. That's encoded by the viral genome. And so if we do this experiment at neutral pH, we get no infection at all. And that pretty much validates the assay because it tells us that there's no infection if the media that the cells are in is at neutral pH. But as we drop the pH, we get this really nice spike of fusion between pH 6 and pH 5, which is exactly what the literature describes as being the pH optimum for membrane fusion by these glycoproteins. So essentially, what's happening here is that the virus sees this acid pH, the GN-GCs dissociate, GC inserts into the target membrane, the virus fuses into the cytoplasm, and then it can express its genes. And we see something very similar whether we're working with Pumila or Andes virus in the VSV system. So we now have this assay where we can actually interrogate viral membrane fusion in the presence of an antibody, and that's really what we're trying to do. So
1 minute.
I only have 1 minute left? Okay, fine. Let me just quickly wrap it up. So basically we can show that we can pre-incubate I probably need like 5 minutes.
Okay.
Absolutely fine.
So we can basically block fusion by pre-incubating with the antibody, and it works at really, really low concentrations. And it also works with the New World hantaviruses, but what's quite interesting is that you need way more antibody to block Andes than Pumila. And that kind of fits with the fact that, of course, this is a Pumila virus antibody, and even though it neutralizes Andes, it's not as potent, right? So basically the potency is much higher for Old World versus New World hantaviruses. And so this kind of got us to thinking about binding affinity. And so essentially what we found is that if you have a bivalent IgG, both the antibody binds fine to both Pumila and Andes, but if you make it into a monomeric FAB, you completely lose binding against Andes virus. So what that tells us is that even though under an avid binding situation, you can get really nice binding and neutralization with Andes, once you get rid of avidity, by eliminating one of the combining sites, you now completely lose binding. And so this basically tells us that affinity is a lot lower for Andes. And we see the same thing by BLI. So just in the interest of time, I'm just going to just cut to the chase here. We wondered if, the difference in, because we saw similar binding avidity with the IgG, we figured neutralization should be the same, even though the monomeric binding affinity is lower. So why is Andes not neutralized as well by this antibody, even when you have an IgG, which is bivalent. So we wondered if there's something else happening here. And so then the thing that really kind of made the most sense was that we know this virus fuses in endosomes at acid pH. So it has to bind the antibody at neutral pH on the outside of the cell, internalize and traffic to late endosomes, and then the pH changes and allows fusion to happen. And so the antibody has to stay bound that whole time. Because if the antibody is released before the virus gets to that compartment, then the antibody is not going to be as effective as blocking fusion because it's not there at the party. And so we asked if acid pH could actually impact this binding interaction directly. And so the answer is very much yes, because as you titrate the pH down, you selectively lose Andes binding while retaining pumila binding. And you can do this in a more appropriate and physiologic way by binding at neutral pH and then dropping, like washing with acid pH to allow dissociation. You do that with pumila, you get this nice acid dissociation curve showing that we can actually see this phenomenon. And you do the same thing with Andes and antibody pretty much falls off at acid pH. So we thought that this difference in binding at acid might really be the driving difference between the neutralization potencies. And so to do this, again, in the interest of time, I'll skip all this, but the mechanics of this, but we did affinity maturation, and we were able to produce a particular variant called 65534 that actually is a much better neutralizer of Andes, but does it without losing its neutralization against pumila. We can also show that if you're looking at, just look at here at the off rate at acid pH, left side of this plot means lower off rate, which means better binding. And so we can see that with these two affinity matured variants, with pumila, they're all about the same as the parent, which is in purple. But with Andes, we see a really nice left shift in the off rate at acid pH with the affinity matured variants, which tells us that we're getting better binding at acid, better retention of binding. So what does this all mean for an animal challenge model? So to really test this, we had to basically reduce the amount of antibody so that we could get breakthrough sort of death with the control antibody, and we can see that really nicely at 0.05 milligrams per animal with the parent antibody. We do the same experiments with the affinity matured version. We can get almost complete protection at this low dose where you get substantial breakthrough. So yes, the affinity matured antibody is actually better against Andes. But the caveat is that we're treating at 3 days post-infection, which is not that realistic in terms of what's happening physiologically. This is actually a slide I stole from one of Pablo's papers showing that there's this pretty long incubation time before people really develop symptoms where they've already been infected, see something very similar in non-human primates. So we really asked, how far can we push this Syrian hamster model? In the Syrian hamster model, which is much more sort of fast, the latest that people have been able to give antibody and protect is day 5. And so we wondered if we could actually go further. And so to do that, Catalina Flores and Andy Herbert's lab did a serial SAC experiment where she could look at the arising of viremia, viral load over time. And we could see this nice peak between days 6 and 9. And so Catalina basically did a walkout study where she didn't give the antibody until day 6 or 7. So here are the controls, the vehicle controls on day 6, all the animals die. And we can get really nice protection still on day 6 with the single dose of the antibody. And then we can do the same thing at day 7. Again, the controls die. And again, we get almost complete protection. So you can actually walk this out as far as day 7 and still get substantial protection. So right now, what are we working on? We're working on how much further we can go with this. We're trying to enhance effector functions, which I haven't talked about at all, but which is actually really important for our Ebola antibodies. We're working with Pablo and others to do developability and manufacturability. We're looking at scale-up. We're trying to get to the point of being able to do proof of concept in non-human primates, more mechanistic studies, and then looking at synergy by combining antibodies, which maybe also, that means you have to manufacture 2 antibodies, which is going to cost twice as much, but it would reduce the risk of escape, and maybe we could reduce the concentrations of these antibodies if there's really functional synergy. So that's something we saw with antibodies against Crimean Congo hemorrhagic fever virus, and we're hoping we can see something like that here. And then we want to apply this knowledge of this new epitope that we've identified to design broadly protective antivirus and vaccines, maybe by steering the immune response to that epitope. And then another sort of interaction I've been having here with Pablo's help is with Alexis Calerhis at Católica, and they're constructing a CGMP facility for vaccine manufacturing, and we're having conversations with him and exploring other options to see if we could find a way to actually manufacture our antibody somewhere in South America for local use. Because right now, we have a cell line producing the antibody, but we have really nowhere to go with it in terms of making clinical grade material. And so with that, I'll just stop and I'll thank this great consortium that I've been really honored to be a part of, that's done all the work that I showed you today. This is just a group picture from one of our annual meetings. I think this is from a couple of years ago, which are really well attended and a lot of fun. And this is my lab. And the two folks in my lab who have done a lot of this work are Eva Mittler, who started my lab as a postdoc 10 years ago and stayed on as a research faculty member, and Megan Slav, who's a postdoc in my lab as well. So with that, I will stop, and if there's time, I'll take any questions.
Questions? We have a couple of minutes.
So this is very, very, very interesting. You're showing a cartoon, no, showing that the viral entrance, it also requires the lysosome.
Mm-hmm.
Have you thought about combining the antibodies with drugs that are FDA approved that target the lysosomal pH and lipid content?
Yeah.
It's a combination therapy.
We haven't done that. We played with that a little bit with Ebola.
PC1 Ebola.
Yeah. I think targeting that pathway always makes me nervous because I feel like a lot of the drugs are kind of relatively non-specific and tend to have lots of other issues. I mean, phospholipidoses and all these sorts of things.
They are not specific, but if you use them for 10 days or 2 weeks.
Yeah, I mean, it could certainly be quite interesting. Something that we've done for Ebola is to make these what we call Trojan horse bispecific antibodies, where one antibody binds to the virus on the outside, and then the other arm of the antibody doesn't work, can target the receptor once the virus is in the endosome. So those kinds of studies could also be interesting for hantavirus. We just haven't done them yet. So.
Thank you very much. Beautiful talk. I was wondering if you could comment more on you started by alluding to the problems. You fast forward, you end up having a beautiful antibody. Mm-hmm. The problems manufacturing them for conditions that are not as frequent as others. So what are and I'm sure you've had already conversations for Ebola and other situations. So how do you see that problem unfolding?
It's purely a problem of money. So for Ebola, the way we were able to partially circumvent that problem was because the US government saw developing an Ebola therapeutic as a priority because of the West African outbreak. So they funded, really all of the preclinical studies, IND enabling studies, and a phase 1 clinical trial. So that was about $75 million. I don't think we need that much, but because this is all funded by BARDA, which is sort of this, which people are probably familiar with. But BARDA has no interest in hantaviruses right now. So that's part of the problem. We've gone to the DoD, Department of Defense, to see if they're interested. They're also potentially interested, but they have more. The problem is that the people the usual suspects that are not companies, like the nonprofit, like government, that would fund something like this for pandemic preparedness or as a public good, they don't find hantaviruses high priority yet. And so that's really the challenge. And so I think we have to be more innovative about how we're going to go about raising money. And that's also why I thought coming to Chile and talking to people and working with Pablo and Alexis and others, maybe there's an opportunity to convince governments, the Chilean government or maybe other governments, that it's in their best interest to provide a bit of money to drive this, at least at this stage, so that we can kind of get past some of these initial blocks. And then once we get going and we have some, we have a GMP cell line, for example, then maybe it gets a bit easier to go to Wellcome Trust or Gates or CEPI and convince them to get us some more money. So I think we're going to have to patch it together. The NIH also has this sort of funded service called preclinical services that will do some preclinical testing. When I go back, I have a call with them and with Larry Zeitlin, who's the CEO of the company that kind of has been developing this with us as part of Prometheus. And we're going to see what they can do to help us. Problem is that they are very slow. So, but we can get it started while we're exploring other options. I mean, in the bigger picture is we're not seeking licensure. I think like trying to get FDA approval or FDA licensure is probably doesn't make a lot of sense because, we've had these multiple conversations that it's going to be quite challenging to do any kind of clinical trial with this kind of therapeutic. But what would be really great is if we could manufacture, if you could manufacture like a few thousand doses, and then just have that, and like if you guys have hantavirus outbreaks, you should have a few thousand doses in the freezer that you can give to people, at least under compassionate use, and then maybe clinicians like Pablo could actually collect data about efficacy, and at least that in that more informal setting. And it would give us an opportunity to like maybe drive towards like a clinical trial. But we're not going to be able to do clinical trials and get approval with all these different therapeutics for emerging infections. We're just going to have to stockpile and have like things in the bank for in case something happens.
And have good registries when you use them to show they work.
Exactly. Yeah. Yeah.
Thank you for the talk. I really like it. I understand the purpose of the I wish you a great success with special situations. But I'm wondering what happened with the antibody response when you use monoclonal antibodies in the animal right now. If you abort the production of natural antibodies?
No. So we haven't done those experiments with the hantavirus experiments in the hantavirus system yet, but what we've done with Ebola clearly shows that in mice and in non-human primates, especially when you do a post-exposure treatment, so you give the animal the virus, and then you come in with the antibody, and the antibody is protective, the animals do mount a protective immune response. But because then you can basically, you can back challenge them with the virus after the antibody has been cleared. So with just a very concrete example, in mice, if you infect the mice and then you come in with the antibody, with Ebola, and you come in with the antibody 2 days later, the mice don't die. And then you wait a month, and then there's no more antibody in circulation, and then you infect them again with the strain of Ebola. They all survive, and they mount an anamnestic response. So at least in a post-exposure setting, we would not expect the natural response to be affected. However, in a prophylactic setting, I don't know what would happen, because if the virus never replicates, then the immune system may not see it, and so you may not boost a very get a very strong response. But given that if you get Andes, I mean, you have a pretty high chance of not having a good outcome. If I were in that situation, I would take the prophylaxis.
All right, thanks, Dr. Chandran. It was a fantastic talk, and now I'll switch to Spanish. Para darle un aplauso al Dr.